Okay, thank you very much. So we move now on with Patrick Frederix. And the title of his talk would be Novel Single Cell Manipulation Approaches to Nanomedicine by FluidFM.
Okay, thank you very much. So we take a large step away from clinical trials. We are now really, I'm going to present a tool for more fundamental nanomedicinal research. And this is a project from a collaboration project. Where is the laser? Yeah, okay. Between Nanosurf and Cytosearch. Cytosearch is a spin-off company from ETH where they developed the microfluidics, the microfluidic part of it. Nanosurf is an AFM manufacturing company. And together we are working on this project. And I want to show you some applications where I see synergies for nanomedical research, and that's data made by our customers. So what is FluidFM? FluidFM is a multifunctional nano-manipulation tool. What does that mean? It means that we have optical control, we have in general the inverted microscope so that we can do normal transmission light fluorescence to study for example, cells. We have position control either by the motorized stage to move the sample, but at a higher accuracy also by an AFM scan head. Force control, the manipulator is mounted on a spring and on a cantilever, which means that if you touch it, the cantilever will bend. This bending can be measured so that you can measure the force, but that you can also bend away from the sample. So rather bend the cantilever than damage the sample. And last but not least, there is integrated microfluidics. So there is a liquid reservoir, and this is about 5 microliter volumes you put in there. And over a tubing, you can either press liquid through a narrow channel out of the cantilever to locally deposit material, or you can also aspire by putting a vacuum pressure, putting an aspiring pressure, either take out material there or pick up large particles at the opening there. I will now go through a few of the examples, and the first example is that you can use with such a hole you can pick up a cell and measure the force that is required to do that. So you first, at pinpointing on a cell, you move the sample to this cantilever, you wait a few seconds to adhere it by the aspiring force, and then afterwards the cell is released from the surface. So the cell that was first there has been detached, and during this process you can measure the force and the energy that was needed to pull that cell away. In the group of Julia Vorholt, Eva Pothoff did a lot of work on structuring surfaces and how does the structuring of a surface change the adhesion behavior of endothelial cells. And this is, for example, important for implants that you put in the blood vessels. You don't want to have blood clogging there, and you want to have to prevent that. You want to have a fast and solid adhesion of the endothelial cells to the surface. So they compared flat surfaces with structured surfaces. The structures had 1 micrometer separation, and the depth was now varied between 100 nanometers and 1 micrometer, and then in addition they had a control with bleb cells. And they see if you structure the surface, the adhesion is largely increased. They combined these experiments also with fluorescence experiments to look at the proteins involved in the focal adhesion points, and other techniques to measure the diffusion of cells on such structured surface. But with FluidFM now you are capable to measure a larger amount of cells, or at least for AFM For AFM, these are larger numbers. If you do normal cell adhesion experiments, you're talking much less cells, or you're talking about much longer periods of time. So this cantilever that you saw here in the experiment, you can push away the cell and pick up the next cell with the same cantilever, which is normally not trivial. So is it only possible for mammalian cells? No, you can also do smaller cells, and here you see two examples, also from Eva Pothoff. She started with yeast, where she compared some mutant yeast with a wild type, and then she studied how the adhesion changes when she brings the yeast cells from 20 degrees to 37 degrees. You see that in the mutant, the adhesion is not increasing anymore, and with the wild type, the adhesion was increasing in time. It can go even smaller, then you don't have a hole at the end. You have a pyramid again with a small hole, and then you can also pick up bacteria and measure the adhesion of bacteria to the surface. And for example, here, study the adhesion kinetics in the short term. So this is what happens in the first minute, where you see that the adhesion clearly is increasing. And of course, bacteria, I mean, to get biofilms, they first need to adhere, and bacterial films are a problem with as sources for infection. What else can you do? And this is as far as we get with counting. We don't count large amounts of particles. What was done in this experiment, that was by Philip Stiefel, he deposited single virus particles on a cell. So he picked a cell, put the cantilever above, put the cantilever on the cell, and because the cantilever is relatively soft, you can really put it on without damaging the cell, and then push out some virus particles. I don't know whether the 4 spots are visible, but in this case there were 4 particles deposited. The virus particles had been modified so that they had a red fluorescent protein on their coating, so that you can nicely see them. There was a second modification on the virus particles, that along with the proteins and enzymes in the early gene expression, a GFP is produced, and with this they could now at the single cell level follow the chance of infection. So here you see this cell, after 7 hours it starts to produce GFP. That means that the early gene expression has successfully started. 2 of the 4 virus particles were left. That gives us also an idea of how many % of the virus particles are entered. Then after 12 hours you see that the amount of GFP has strongly increased, and now you see that also new variants have been assembled in the late phase. So what they see, if they deposit a single virus particle, you have about 10% chance of infection to come to this stage. If you have 2, it's not about 20%, you're talking about 35%. So it's more than deterministically increasing with the number of deposited viruses, and at 4 or 8 particles you have 100% chance of infection of the cells. So far that was deposition on top of the surface, but if you take again a different shape, now you take a cantilever which has a hole at the side of the tip, and here's the sharp tip of a normal cantilever. The cantilever itself is again hollow so that you can push through liquid, and this sharp tip you can push into a cell, and not even into the cell, not only in the cytoplasm, you can also go into the nucleus so that you can do intranuclear injection into a cell. So they had a marker in dextrin with a fluorescent marker that you can put into the nucleus. Like with the large molecule on top of it, it cannot leave the nucleus through the pore complexes to the nuclear envelope. That means that you can put in the cantilever, you can take it out, and the nucleus envelope and the cell membrane are recovering from this injection. Otherwise, the probe would not be localized at the nucleus. There was again a GFP plasmid along with this dextrin. This gives also a measure of the concentration, and then they could study the same cell for days, several days, to see what happens with that cell. So this is that optical control that you have. You can do the manipulation with the AFM, but afterwards you can still study the same cells for multiple days. And with this method they got here about 40% transfection efficiencies in these, if I'm correct, HeLa cells. A very recent paper from Dario Ostola, he's in the group of Nanosurf at the ETH, is to use the FluidFM with the hollow cantilevers for patch clamping. And the goal of patch clamping to measure ion currents through the cell membrane, and the conventional way is to have the glass pipette. And now the idea is to get, can we do the same with an AFM tip, so that you have there again the hollow cantilever opening at the end, an electrode in the reservoir, second one there, and can I now measure the whole cell conductivity if I break the membrane there? So they tried this on sodium channels and did for a first proof of principle, so that they can get there the step responses of the current as function of the steps. And that is one experiment, and what is more interesting for me is that they could do it actually on beating hard cells. These cells are tricky to do with a glass cell. The cell is moving. That means that you have to take care that you don't lose the sealing. With a glass pipette, you most likely lose the sealing or you break the cell. The cantilever could actually measure a lot of beatings in sequence, and you see there in black the current response, and there the bending of the cantilever measured here as a force, giving the mechanical movement of the cell. So you can now measure the electromechanical relationship of the hard cells and actually measure on living beating hard cells. So what I hope that I could give you in this short presentation is that FluidFM is a forgiving nanomanipulation tool. You can go onto a cell without damaging it, and you can follow that cell in time. For patch clamp, you don't lose the sealing if you once you have a contact. And as you saw from the few applications that I showed, there is a large diversity of applications. So there is the force spectroscopy, there is patch clamp, there is virus deposition, injection, and with this I would like to show you with one small last application that you can also modify surfaces with the FluidFM, and that was a movie made also at ETH with Raphael Grüter in the group of Zambelli and Nanosurf. And with this I would like to thank you for your attention.
Thank you very much, Patrick. Any question to his lecture in information he provided? Yeah, please. You have a little bit longer way to the microphone.
Very nice talk. My name is Melanie Cookie. I come from EMPA St. Gallen. And my question is, can you functionalize your AFM tip with nanoparticles so that you can measure the adherence of nanoparticles to the cell surface? Would it be possible?
Yes. So what I would do, in principle, you can do it with standard cantilevers. Then, of course, once the cantilever is contaminated, you have to take it out. What we are considering is that you take larger particles and coat these larger particles with nanoparticles. And with the hollow cantilever, you can aspire a nanoparticle to the opening, then do the experiment with the nanoparticles on it. And then when you see, okay, my bead is contaminated, you just put a positive pressure, get rid of the bead, pick up the next bead, either with the same coating to increase your statistics, or with a different coating to measure response on different types of nanoparticles, so that you can there have a higher throughput to study the particle interaction.
Okay, thank you very much.
Any further comment, question to Patrick?